I think for me it's fascinating because you have a very strict code, a very strict program how the brain develops. So every day, every week, the neurons from the group of stem cells, they know where to go and when to go and which direction to go. So this is a beautiful program, a beautiful code, and if you understand this code, then you can translate it to every day of the development, what can go wrong if there is a damage in a certain day. In the brain we see loss of neurons. This is one of the most important definitions of neurodegenerative disease, that the neurons are lost. So in anatomical areas where we expect from normal brains to be a lot of nice neurons, we don't see these neurons, they are lost. And additionally there is a reactive change, and these glial cells, the astroglial cells, then take the place of the lost neurons, and we call it astrogliosis. So this is what we see. In addition, there is a problem with the protein processing of the neurons, and which means that there is accumulation and deposition in the brain of pathological proteins. That is a very characteristic feature of neurodegenerative diseases, and this is what we can examine in the microscope, and we can classify and group the neurodegenerative diseases according to which protein and in which cell, which anatomical area is deposited. The focus of our project is Alzheimer's disease and related dementing illnesses. Dementing illness means cognitive decline, when somebody loses the normal human cognition gradually, and this is what we are examining. Our focus is to define the earliest changes, what goes wrong in an Alzheimer's disease brain. So we usually say, or researchers say, that Alzheimer's disease is a lifelong story. It depends on how somebody is educated, how a genetic background looks like of an individual, how protected his or her neurons are. So it's a lifelong experience. Usually many years before the patients go to the doctor, the neurons already die and the contacts between the neurons are lost. And these proteins start to accumulate and deposit in the brain. However, our brain has a reserve capacity, and it compensates. And this is why we don't see for a long time that there's something problem. Only when there is a threshold, and when this accumulation of proteins and loss of neurons reaches this threshold, then the patient goes to the doctor. In human development, when the brain develops, there's a certain codes and program how it goes. There are several pathways. So they say that this is a pathway, this neuron has to go here, the axons guide this, and the glial cells guide these neurons or stem cells to go to the end place where they will then stay, and in the adult brain they will be there. These pathways and these are very important pathways, because they tell a neuron or a stem cell to be a neuron, to go to a certain place and to make a function. So if you change this program in a developing brain, then maybe this neuron will start to go somewhere but will not reach it, or will not make the best contacts with other neurons. And when somebody gets older and starts a neurodegenerative disease, if these have not been well developed, the contacts, then this can lead to an earlier appearance of symptoms as compared to another individual where these contacts are well developed. We try to find if really in individuals who are prone to get Alzheimer's disease, we can go back and find a problem during the development of a human brain. So whether really in patients with Alzheimer's disease the neurons did not go to the right place, or if they went to the right place, they did not really develop all the good contacts that they need. This consortium is based on many teams, and the major aim was a multidisciplinary approach that we complement each other. So this means that we bring many different way of thinking into this project. So researchers who use different technologies, different methods, and think of the human brain in a different way. If we think of the human brain and do research in a different way, then when we meet, we complement each other. And then of course, we needed experts who are experts in the developing brain. We have experts who are experts in the aging brain. And we have experts who are experts in the basic processes of what goes wrong in the brain, genetic or epigenetic. And then, of course, research needs always experimental models. So we needed groups of researchers who can help to develop experimental models, animal models, which can be used as a model for human disease. If we could do this project for 20 years more, I think that we would be really able to find those pathways during the development which go wrong, and for this reason somebody 80 years later develops or more prone to get Alzheimer's disease. I think if we could go more further, then we could find the genetic protective and risk factors in more details. I'm sure that we will already find during our project, but because the human genome is so complicated and so large, if we could do it for more time, we could evaluate more individuals, and of course we would have a more chance to map the variability what is in the human population.